Welcome back, everybody. Day 40, can you believe it? 40 days of diving deep into cellular kinase receptor therapies. And honestly, I'm still learning so much.
Me too. It really highlights how much is happening in this area.
Definitely. We've covered just how crucial these receptors are, in cell signaling, making sure everything runs smoothly from growth to development.
And of course, when they go wrong, that's where we see diseases like fibrosis and even cancer rearing their ugly heads.
Right, and that's where today's focus comes in, right?
Exactly. We'll be zeroing in on the TGF-beta and BMP pathways. They're both part of this big kinase receptor family.
And they're like the hot targets for new therapies, right?
Absolutely. But before we get into the nitty-gritty of targeting them, remember earlier we were talking about microdomains? I think it's worth revisiting that because it changes the way we even think about cell signaling.
Yeah, I remember finding that whole concept really mind-blowing.
It really challenges the old way of looking at things. For so long we pictured signals spreading evenly throughout the cell, like when you drop dye in a glass of water.
Right, like it just diffuses everywhere equally.
Yeah, but that's not actually how it works. Cells are way more organized. It's more like thinking of a city with different neighborhoods, each specializing in specific activities.
Okay, I like that analogy.
Yeah.
So how do these cellular neighborhoods or microdomains actually work?
So imagine a signal, like cyclic AMP, that's supposed to turn on a specific enzyme, protein kinase A or PKA. In the old global signaling model, that cyclic AMP would just float around and activate any PKA it bumped into.
Right, kind of like a citywide announcement system where everyone
Yeah.
gets the message.
Exactly. But that's not how cells do it. Instead, these microdomains create little pockets of PKA that are localized, and they only respond to signals within their specific zone.
So it's more like targeted messages delivered only to certain neighborhoods. That's super efficient.
Exactly. It's about keeping things controlled and making sure the right signals go to the right places.
But how do they actually do that? How do cells keep things so organized?
That's where A-kinase anchoring proteins, or AKPs, come in.
AKPs, right. I'm starting to remember now.
These proteins are basically the scaffolding. They bring PKA to specific locations and hold it there.
So they're like the city planners, making sure the right resources are in the right neighborhoods.
Precisely. And here's the kicker. There are over 50 different AKPs in humans, each with its own preference for different types of PKA.
Wow, so that's a pretty complex zoning system. Makes you wonder if this microdomain thing happens in other places too.
Oh, absolutely. In fact, you brought up thyroid hormone receptors earlier, or TRs as they're known. They're a perfect example of how microdomains make a big difference in how cells respond, even to the same signal.
Oh, yeah, that's right. We were discussing how they can act as both gene silencers and gene activators. How does that fit in with this microdomain idea?
Well, you see, different tissues in our body make slightly different versions of these TRs.
Oh, really? So different flavors of the same receptor?
Exactly. And each of these versions, these isoforms as they're called, they have their own unique quirks, their own ways of interacting. So even though it's the same thyroid hormone floating around, the effect it has will depend on which TR isoform it encounters in a specific microdomain.
That's fascinating. With like different dialects of the same language. The basic message is the same, but the interpretation can change depending on who's listening.
I like that. Perfect analogy. It really adds another layer of complexity to the whole signaling process.
It really does. But, stepping back a bit, why should we even care about these TRs in the first place?
Good question. They're actually super important because when things go wrong with TRs, it can lead to some serious health issues.
Like what? What kind of problems can happen?
Well, for example, mutations in one type of TR beta specifically, it can cause something called resistance to thyroid hormone, or RTH syndrome. Basically, the cells become less responsive to thyroid hormone.
And what does that actually look like for someone with RTH? What kind of symptoms do they experience?
It can manifest in a whole range of ways, from attention problems to growth issues to even hearing loss. It's really quite varied.
Wow, so that really shows just how important these receptors are for normal development, even if we don't always think about them.
Absolutely. And unfortunately, it's not just RTH. There are other examples of what can go wrong with these TRs. There's actually a mutated form of another type of TR alpha this time, and it can actually contribute to cancer.
Wait, seriously, a messed up TR can actually help cancer cells grow.
Yeah, it's this mutated form called the VRB oncogene, and it's constantly silencing genes no matter if thyroid hormone is present or not. It's like a switch that's stuck in the off position.
And I'm guessing that throws a wrench in the whole gene regulation system.
Exactly. And that kind of disruption can actually fuel uncontrolled cell growth, which is a hallmark of cancer.
That's pretty scary stuff.
I
But I'm hoping researchers are looking into this, trying to figure out how to fix these problems.
Oh, absolutely. They're using animal models to study the different roles each type of TR plays. That's going to be crucial for developing targeted therapies to tackle these dysfunctions.
That's good news, at least.
hmm
And it seems like every time we uncover a new layer of complexity in these pathways, it opens up new possibilities for treatment.
You got it. And speaking of uncovering new layers, we need to talk about this protein called NCOR.
NCR, I haven't heard of that one.
It stands for Nuclear Receptor Core Pressor.
Okay, that sounds important, but also complicated. What does it do?
Well, it actually works alongside those TRs we were just talking about, helping to silence genes when thyroid hormone isn't around.
So it's like the opposite of an activator, the brakes to the accelerator, so to speak.
Exactly. But here's the thing, and this is what's so fascinating. When researchers messed around with NCOR levels in mice, they found something really unexpected. They increased NCR in the livers of these mice, and guess what happened?
I'm going to guess it messed with their thyroid hormone balance.
That's a reasonable guess. But actually, they saw an increase in cell proliferation, meaning the cells started dividing more rapidly.
Wait, hold on. I thought NCR was supposed to keep things quiet, not make cells go into overdrive.
I know, right? It's a total head scratcher. And it suggests that NCR might be messing with some other pathways that normally control cell growth. Specifically, researchers think it might be interfering with the Mike-Max-MAD pathway and the E2FRB pathway.
Okay, those names are a mouthful. Can we break down what those pathways actually do, like in simple terms?
Of course. Imagine MyTink as this super powerful growth promoter, like the gas pedal for cell division. And usually there's a braking system involving MAD and MAX, and they use NCR to help suppress MyMax activity.
So it's like a constant tug of war with MyRAC pushing for growth and Mad Max and NCR trying to hold it back.
That's a great way to put it. But when you have too much NCR around, it seems to disrupt that delicate balance, like cutting the brake lines and letting MyPrec push that gas pedal to the floor.
Okay, I can see how too much NCR could have some unintended consequences. But what about that other pathway, the E2FRB one? What does that do?
That one's also a major player in controlling the cell cycle. Think of RB, or retinoblastoma protein, as the gatekeeper, making sure cells don't move on to the DNA replication phase until they're good and ready.
So RB's like the checkpoint, making sure everything is in order before the cell starts copying its DNA.
Exactly. And E2F is like the construction crew, ready to build new DNA once they get the green light. And you know what? RB actually recruits NCR to help keep those E2F genes quiet until the right time.
So NCR is playing both sides then, helping to control mycotic and E2F. That's a lot of responsibility for one little protein.
It is. And this research suggests that too much NCR might be interfering with RB's ability to control E2F, which could potentially lead to more cell division.
Wow, this is getting pretty complex.
Yeah.
But I'm starting to see just how much these microdomains rely on a delicate balance. It's not just about having the right proteins, it's about having the right amounts and making sure they're interacting in the right ways.
It really is like a beautifully choreographed dance. And this whole concept of microdomains is really changing how we understand how cells work at a fundamental level.
You know, it's easy to get lost in the weeds with all this talk of microdomains and signaling pathways. But remember, way back at the beginning, we were talking about actually manipulating these pathways, right?
That's right. We did start there. But, understanding the fundamentals of how these signals work, especially the whole microdomain concept, it's crucial if we want to figure out how to target these pathways effectively.
Okay, that makes sense. So just as a reminder, what makes the TGF-beta and BMP pathways so interesting when it comes to developing new therapies?
Well, they're both part of this big superfamily of signaling pathways called the transforming growth factor beta superfamily. And they're basically essential for all sorts of things, like controlling cell growth, deciding what type of cell each cell becomes, and even making sure tissues and organs form correctly.
Okay, that sounds pretty important.
It is. But what makes them especially attractive for drug development is that they're involved in a whole bunch of diseases.
Okay, now we're talking. So what kind of diseases are we talking about here?
Well, one of the big ones is fibrosis.
Fibrosis, right. I've heard of that, but can't quite remember what it is.
It's basically when you have this excessive buildup of scar tissue in an organ. It can happen in the lungs, the liver, heart, kidneys, pretty much anywhere.
And that's a problem because
all that scar tissue can really mess up how the organ works. It can lead to damage, and eventually the organ might even fail.
that sounds pretty serious.
Okay.
And what do TGF-beta and BMP have to do with all this scarring?
Well, they actually have this kind of dual role, which is what makes them so interesting. Early on, after an injury, they can actually be helpful. They promote wound healing and make sure tissues repair properly.
So they're like the first responders rushing in to fix things up.
Exactly. But here's the catch. If their activity isn't kept in check, things can go awry. They can actually end up promoting too much scar tissue formation, which leads to fibrosis.
So it's kind of a double-edged sword. Helpful in the right circumstances, but too much of a good thing can actually backfire.
Exactly, and that's precisely why researchers are working so hard to figure out how to fine-tune their activity. One way they're trying to do that is by blocking the signals from specific TGF-beta or BMP ligands.
Remind me again, what are ligands? It's been a while since Bio 101.
No worries. Ligands are basically the signaling molecules that bind to those receptors we were talking about, which kicks off the whole signaling cascade.
Right, so it's like intercepting a message before it gets to the intended recipient.
That's a good way to think about it. And there are already a bunch of inhibitors and antibodies in development that target specific ligands or receptors in these pathways.
So they're basically trying to shut down those signals that lead to excessive scarring.
That's the idea. By blocking them, researchers hope to stop or even reverse the whole fibrotic process.
That sounds really promising. But I have a feeling it's not as simple as just shutting down those pathways completely. There must be some downsides, right?
You're absolutely right. Remember, these pathways are involved in a lot of normal processes too, and they're all interconnected in complex ways. Blocking one part can sometimes have unintended consequences. We call these off-target effects.
Ah, yes, off-target effects. Like the unwanted side effects that can sometimes derail a promising new drug. What kind of off-target effects have researchers seen with TGF-beta and BMP inhibitors?
Well, because these pathways are so important for normal development and how tissues function, blocking them can sometimes disrupt those processes. For example, TGF-beta has a hand in regulating the immune system. If you block it, you might increase the risk of infections or autoimmune diseases.
So it's like a delicate balancing act. You want to block enough of the signal to prevent the fibrosis, but not so much that you end up compromising the body's other essential functions.
Precisely. And that's one of the biggest hurdles in developing effective therapies that target these pathways. Researchers are constantly working to make these inhibitors and antibodies more specific, trying to find that sweet spot where they can effectively treat the disease without causing a bunch of unwanted side effects.
It sounds like a long road, but the potential benefits are huge. I mean, if we can harness the power of these pathways, it could revolutionize how we treat fibrosis and so many other diseases.
Absolutely. And speaking of other diseases, let's not forget about cancer. Remember that VERB oncogene we talked about earlier? That's just one example of how these pathways can go haywire and contribute to cancer.
Right. It's like these pathways have a dark side.
Mhm.
They can help with growth and repair, but if cancer cells hijack them, they can actually fuel uncontrolled growth.
Exactly. And that's why researchers are also looking at targeting these pathways in cancer. Some of the same inhibitors and antibodies being tested for fibrosis are also being investigated as potential cancer therapies.
That makes sense. If you can block the signals that are driving cancer cell growth, you might be able to halt the tumor or even shrink it.
Exactly. But as with fibrosis, it's not that straightforward. Cancer cells are notorious for developing resistance to treatments, so researchers are always trying to stay ahead of the game, coming up with new and better ways to target these pathways.
It sounds like an ongoing arms race between scientists and cancer cells.
You could say that. But thankfully, scientists are making progress. There's a lot of buzz about these new immunotherapies that can help the body's own immune system recognize and destroy cancer cells.
I've heard about those. They sound really promising.
They are, and some researchers are combining these immunotherapies with inhibitors of TGF-beta and BMP pathways to try and boost their effectiveness even more.
Wow, that's incredible. So they're basically helping the body fight back and at the same time blocking the signals that protect the cancer cells from the immune system.
Exactly. It's a powerful combination and really shows just how creative and dedicated researchers are when it comes to fighting this disease.
It's definitely inspiring to see how far we've come in understanding and targeting these pathways. But I'm curious, are there any other frontiers beyond fibrosis and cancer? Are there other areas where TGF-beta and BMP might hold promise for new therapies?
You know, you bring up a great point. We've focused a lot on the disease side of things, but there's another exciting area where these pathways could really make a difference: tissue regeneration.
Oh yeah, I vaguely remember reading something about that.
Think back to what we were discussing about their role in development, how they act like master architects guiding the formation of organs and tissues in the embryo.
Right, they're like the blueprints for building a whole organism.
Exactly. And now researchers are trying to figure out how to harness that same power to repair or even regenerate damaged tissues and organs in adults.
Wait, are you saying we might be able to regrow damaged organs using these pathways? That sounds like science fiction.
It might sound futuristic, but it's becoming increasingly plausible. Researchers are using TGF-beta and BMP proteins to guide stem cell differentiation.
Stem cells, those are the blank slate cells that can turn into any type of cell in the body, right?
Exactly. So by exposing stem cells to specific TGF-beta or BMP proteins, they can coax those stem cells into becoming the specific types of cells needed to repair or regenerate a particular tissue.
So it's like giving the stem cells a blueprint and a construction crew to build new tissues.
That's a great analogy, and it opens up incredible possibilities for treating a whole range of conditions, from spinal cord injuries to heart disease.
This is mind-blowing stuff. We've gone from understanding how cells communicate to actually manipulating those conversations to potentially heal and rebuild entire tissues.
It's a testament to the power of scientific curiosity and the dedication of all those researchers out there pushing the boundaries of what's possible. By figuring out how these complex signaling pathways work, we're opening doors to therapeutic avenues that we could only dream of before.
I'm truly in awe of the intricate beauty of these molecular mechanisms. But after all this discussion, my brain is feeling a bit full.
I can understand that. It's a lot to take in.
Maybe we can take a moment to recap some of the key points we covered today.
Definitely. So we started with that puzzle about how the same cyclic AMP signal could lead to different outcomes in cardiomyocytes, which led us to the concept of microdomains, those localized signaling zones within cells.
Right, and then we dove into the crucial role of AKPs in organizing those microdomains.
And from there, we explored how this concept helps us understand why thyroid hormone receptors can be both gene silencers and activators, and how the protein NCR can unexpectedly influence cell proliferation, adding even more complexity to the whole microdomain story.
And we wrap things up by coming full circle back to those TGF-beta and BMP pathways, discussing how they contribute to fibrosis and cancer, but also their potential in the exciting world of regenerative medicine.
It really is fascinating how these pathways can do so many different things depending on the context, promoting healing on one hand and driving disease on the other.
Absolutely. And what's even more inspiring is to see how researchers are developing all these innovative strategies to target these pathways, giving us hope for treating so many different diseases.
It's a remarkable field and highlights just how important it is to keep exploring and asking questions.
This deep dive has been a wild ride, with so many twists, turns, and amazing discoveries. I feel like I've gained a whole new appreciation for the world of cell signaling.
It's been a pleasure exploring it all with you. And to our listeners, don't forget, the journey doesn't end here. There's always more to learn and more mysteries to unravel.
Absolutely. Keep those questions coming, folks. Stay curious and keep diving deep into the wonders of the molecular world. Until next time, happy exploring.
Oh, absolutely. There are, lots of areas to explore. Now that you mention it, I do remember reading something about TGF-beta and BMP pathways being involved in tissue regeneration.
Oh, yeah. Like, what does that even mean? I mean, are we talking like Wolverine-style regeneration here?
Haha. Not quite Wolverine levels, but it is pretty amazing stuff. Remember we talked about how these pathways play a big role in tissue development, like they're the master architects, right? Guiding the formation of organs and tissues way back when we're just embryos.
Right, right. They're like the blueprints for building a whole person.
Exactly. And now researchers are trying to figure out how to tap into that same power, but this time to repair or even regenerate damaged tissues in adults.
Wait, hold on. Are you saying we might one day be able to regrow damaged organs using these pathways? Well, that sounds like something straight out of a sci-fi movie.
It might sound futuristic, but it's getting closer to reality all the time. Scientists are already using TGF-beta and BMP proteins to try and stimulate stem cell differentiation.
Stem cells, right. Those are the ones that can basically turn into any type of cell, aren't they?
Exactly. They're like blank slates. So by exposing those stem cells to specific TGF-beta or BMP proteins, scientists can nudge them, guide them into becoming the specific types of cells that are needed for tissue repair.
So it's like giving them the blueprints and the construction crew all in one package.
That's a great way to put it. And the potential is just incredible. We could be talking about treating things like spinal cord injuries, heart disease, all sorts of things that are currently really difficult to treat.
This is mind-blowing stuff. I mean, we've gone from understanding how cells talk to each other to actually manipulating those conversations. And now we might be able to use that knowledge to heal and even rebuild entire tissues.
It's pretty amazing, right? It really shows you the power of scientific curiosity and the dedication of all those researchers who are out there pushing the boundaries by unraveling these incredibly complex signaling pathways. We're opening doors to possibilities that were once unimaginable.
I'm seriously in awe of all this, like the sheer beauty and intricacy of these molecular mechanisms. But I gotta say, my brain is feeling a little full right now.
I hear you. It's a lot to take in. Even for us, it takes a while to wrap our heads around all of it.
Maybe we should take a step back for a minute and just like recap some of the key things we've talked about today.
Sure, good idea. So we started with an interesting puzzle, right? How the same cyclic AMP signal could have totally different effects in cardiomyocytes.
Right, right. And that led us to that whole concept of microdomains, those little pockets of signaling activity within the cell.
And then, of course, there are those AKPs, the organizers of those microdomains. We can't forget about them.
Absolutely. And then we talked about how all of that helps us understand why those thyroid hormone receptors can be both gene silencers and activators.
And don't forget about NCRR and its surprising role in cell proliferation. It just goes to show you how much complexity there is within these microdomains.
It's amazing. And then finally, we circle back to those TGF-beta and BMP pathways, talking about their roles in fibrosis and cancer, but also their potential for regenerative medicine.
Yeah, it's pretty remarkable how these pathways can be involved in so many different things, promoting healing in some cases and driving disease in others.
It really is. And what's inspiring is that researchers are finding new and clever ways to target them. It gives us so much hope for finding new treatments for a wide range of conditions.
It's a truly exciting field, that's for sure, and it just highlights how important it is to keep asking questions and pushing the boundaries of what we know.
This deep dive has been a wild ride, and I've learned so much. The world of cell signaling is truly mind-blowing.
I've enjoyed exploring it with you, and to our listeners, remember, the journey doesn't end here. There's always more to discover and more mysteries to unravel.
So true. So keep those questions coming, everyone. Stay curious, and don't stop diving deep into the amazing world of molecular biology. Until next time, happy exploring.
It is. It really is. And, speaking of other areas, we've talked a lot about, the disease side of things, but these pathways, they also hold a lot of promise for something completely different, something really exciting, tissue regeneration.
Tissue regeneration.
Yeah.
Yeah, I think I remember reading something about that, but it was kind of, over my head at the time.
Well, think back to what we were discussing about their role in, in development, how they act as these master architects, right? Guiding the formation of organs and tissues way back when we're just embryos.
Right, right. They're like the blueprints for building a whole person.
Exactly. And now researchers are, they're really trying to figure out how to harness that same power, but this time to repair or even regenerate damaged tissues in adults.
Wait, so you're saying we might one day be able to regrow damaged organs using these pathways? That sounds like straight out of science fiction.
I know. It does sound futuristic, but it's actually becoming more plausible all the time. Scientists are using TGF-beta and BMP proteins to try and stimulate stem cell differentiation.
Stem cells, right.
Yeah.
Those ones that can turn into any type of cell.
Exactly, like blank slates,
Yeah.
So by exposing those stem cells to specific TGF-beta or BMP proteins, scientists can, they can kind of nudge them, guide them into becoming the specific types of cells that are needed for tissue repair.
So it's kind of like giving them the blueprints and the construction crew all in one,
Exactly. That's a great way to put it. And the potential is just incredible. We could be talking about treating, things like spinal cord injuries, heart disease, all sorts of things that are really difficult to treat right now.
That's amazing. It's like we've gone from just understanding how cells communicate to actually manipulating those conversations. And now we're talking about actually healing and rebuilding tissues.
It really is. It's really remarkable, and it just goes to show the power of scientific curiosity and the dedication of all those researchers out there pushing the boundaries, right? By unraveling these complex signaling pathways, we're opening up all these possibilities, things that we never even imagined before.
It's incredible. The whole, just the sheer beauty of it all and the complexity of these molecular mechanisms, it's mind-blowing.
Yeah.
But I got to be honest, my brain is feeling a little full right now.
Yeah, I hear you. It's, it's a lot to take in.
Maybe we should take a step back for a second and kind of recap some of the key things we've talked about today.
Sure, that's a good idea. So We started with that puzzle about the same cyclic AMP signal leading to different outcomes in cardiomyocytes, which led us to the concept of microdomains, those localized signaling zones within cells.
Right, right. And then we talked about those AKPs, those like the organizers of those microdomains.
Right. And then we talked about how, that helps us understand why thyroid hormone receptors can be both gene silencers and activators, and then how NCOR, that protein, can influence cell proliferation, adding even more complexity to those microdomains.
And then, we circled back to TGF beta and BMP, talking about, fibrosis, cancer, and then, that exciting potential for regenerative medicine.
It really is fascinating how these pathways can do so much. They can promote healing, but they can also drive disease.
Yeah. Yeah, it's incredible, And what's really inspiring is seeing how researchers are coming up with all these strategies to target these pathways.
It's a really remarkable field, and it just goes to show how important it is to keep asking questions and to keep learning.
Absolutely. This whole deep dive, it's been a wild ride. I feel like I've
Yeah.
I've learned so much about, cell signaling, and it's truly mind-blowing.
Well, it's been a pleasure, exploring it all with you. And to our listeners out there, don't forget, the journey doesn't end here. There's always more to learn, more mysteries to unravel.
That's right. So keep those questions coming, folks. Stay curious and keep diving deep into, the amazing world of molecular biology.
Yeah.
Until next time, happy exploring.